Okay, this is the Vertebrate Zoology Lab 1, Origin of the Vertebrates and Development. Stations 1, 2 and 3 at the moment deal with the origin of the chordates. We begin with a hemichordate, which we could probably jettison, but the link here is that we have a stomacord, the pharyngeal slits, and also, to some extent, a dorsal nerve cord, but they've also got a ventral nerve cord. So the idea is they share some traits in common, although we don't know if they're fully homologous or not. Then we have our Urochordata. These guys have the 5 chordate synapomorphies, but of course the larva has all 5 as a filter feeder, but the adult actually jettisons 3 of those. Those associated with movement becomes a sessile filter feeder, so just as a urostyle in the pharyngeal basket. Okay, we then move over to the Cephalochordata. Cephalochordata have all the 5 chordate synapomorphies and retain those into adulthood. We look at these in some detail about their filter feeding method and about the pharyngeal basket. We also look into a little more detail around the pharynx, and we start to look at some of those features, specifically the endostyle, which is a mucus production center still to facilitate feeding. And then in some of them, we can see a Hatchek's pit. It might be in this one. Let's just see. Whoa. Yeah, the Hatchek's pit is up the top of the pharynx there, with the endostyle at the bottom. I'm just not very stable right now. And so the Hatchek's pit and the endostyle collectively are essentially exaptations when they become the pituitary and the thyroid gland. Hatchek's, pituitary, endostyle, thyroid. Okay, we're going to jump a few stations now. We're going to go to station 9 and 10. So station 9 is showing a little tiny larval cephalochordate, but then it shows us a cephalochordate again here under this microscope. And over here we have a larval lamprey, the amocoete larvae. And so really it's just to illustrate the similarity between those two forms. Of course, vertebrates become much more complex than their ancestor, so we develop to an adult larvae through the sort of broad suite of traits for enhanced mobility, the cranium and tripart brain, the vertebrae, and a suite of organ systems and endocrine systems within that too, and a much larger organism. So that's that link to the vertebrate evolution. I then flip to station 4, where we have a sequence of different vertebrate taxa represented, and really the rationale here is the ability to differentiate these taxa by their defining features. So we've got a jawless fish. We then move into the gnathostomes. We have chondrichthyes for everything else. So chondrichthyes as one branch of fishes. Actinopterygii as another branch of fishes. And we don't have a Sarcopterygii, but we do have the tetrapods, which of course were derived from them, with our amphibians. And then we have the amniotes with our turtles, lizards and snakes, archosaurs to birds, and then the mammals as the other lineage within the amniotes. We would ideally see differentiating features. This one's a bit tough. It's lost its bone and it's now just cartilage. These guys have got ray fins, which is very visible there. These guys had fleshy fins. Now it's a tetrapod with out keratinized skin because it's an amphibian, so it's breathe through the skin. These guys have the keratinized skin. We have the scutes and a test of a turtle or a tortoise. We have the scales of the lizards. We have the feathers of the bird. We have the fur mammary glands of a mammal. Okay, now for development. Big toughie here. Okay, we start with a discussion about life histories, going from many small eggs in an animal with high mortality, like early fish, through to amniotes, which have much higher survival and therefore a much bigger egg. We see the same in teleosts, which have long lifestyles as well. And so this necessitates different forms of development to cope with those much larger amounts of yolk in the egg. And so we talk about a transition from an early microdocythal egg, which has holoblastic cleavage all the way through, towards an egg with yolk distribution located beneath the animal area. So you have a vegetal and an animal area, and also an inability for cleavage to continue all the way through. So the amphibian ends up with the unequal holoblastic cleavage, which can go all the way through, but only just. and you have the discoidal meroblastic cleavage of the chicken or other amniotes, and that has also evolved in some fish like teleosts. Okay, we then look at the mammal, which has gone back to being this tiny little microluciferal egg with holoblastic cleavage, and we'll talk about that later because they don't use a yolk, they use a placenta. Okay, introduction to blastulation. We show the consequences for the different types of cleavage with a circular blastula, an unequal blastula, a discoidal blastoderm or blastodisc for birds, and then this blastocyst for mammals. And here you can see it with the epiblasts and the hyperblast internally. Okay, station 8 is all about showing early development in a simple deuterostome. So we look at the echinoderm and its stages of development with the slides and simple gastrulation in an echinoderm. It's really just a segue for the major stages before diving into the amphibians. We do unequal holoblastic cleavage to produce our blastula. We then do gastrulation, emphasizing the grey crescent with sperm entry opposite, causing the animal cytoplasm in the external region to sort of rotate towards that point, thereby defining an anterior and posterior axis and the lateral axis, the dorsal ventral. The dorsal lip of the grey crescent that's revealed is the location of where the gastropore enters. That's the significance of that grey crescent. The gastrocele, when forming, displaces that internal blastocele, and we differentiate our three germ layers, ectoderm, mesoderm, and endoderm, as we see that involution of the gastrocele. That's all covered in these images here. We then reach neurulation, and of course, with a gastrocele running the length of the body, it can then induce the above mesoderm to form a notochord, and that notochord can then induce the above ectoderm to form a neural tube. And as that happens, we see the neural crest cells forming as well. So they're of type of neuroectoderm. And in fact, we have neurogenic placodes forming on the surface. So together with the three germ layers, the neural crest and neurogenic placodes, we can then further undertake organogenesis of those germ layers into the organs of the body, and we see full embryogenesis. But were it so simple, it would be an easy lab. Now we have to go over to the amniotes. Now they're living on land, so they have to have extra embryonic membranes to support waste, a yolk sac. The waste is allantois, a yolk sac for nutrition, a amnion to protect the baby, and then a chorion for respiration with the shell on the outside. So this is going to necessitate components in development that become the extra embryonic membrane and some that become the embryo. And so with our giant amount of yolk for these very large eggs that live on land that are for high long life animals, we have a discoidal cleavage confined to a teeny little area on top because we cannot do cleavage all the way through. This disc divides and actually generates a upper level of hyperblast and a lower level of epiblast, not visible in that particular slide, but conveniently someone has left me one of their lab handouts here. And so within this, if I skip through, aha, here we are. We can start to see this process. So this one shows us what comes about as the blastulation process to generate our upper epiblast and lower hyperblast. And this shows then fate with the primitive streak forming, essentially the length with a Henson's node at one end to become the head, and then our mesoderm and endoderm descending in, and the endoderm pushing the hyperblast to the sides. So the epiblast forms the three germ layers and the embryo. The hyperblast will go and form our extraembryonic membranes. Mesoderm will differentiate. Eventually it's going to form a notochord and then induce neural tube formation. Eventually the sides of this plate actually pinch inwards to generate the gastrocele and then the primitive heart from the mesoderm in these areas. Okay. We then follow a suite of microscopes showing some organogenesis from our different germ layers, emphasis being really on a tripartite brain that then differentiates to a 5-part brain, and also on the somites, which form the muscles and parts of the skeletons. But really we could choose a whole suite of different tissues, and that's covered better in lecture. Nevertheless, somites and the tripartite 5-part brain are quite useful. finally, we hit mammals. Here, of course, we got the ancestral state, but it's complemented by the changes towards having a placenta. So as a consequence, we go and lose all that yolk and end up with our microluciferal egg. It is isoluciferal, but microluciferal egg with a holoblastic cleavage, and it generates a morula and then a blastocyst. And the blastocyst is a ball of cells with an inner cell mass that differentiates to a epiblast and a hyperblast there in the yellow and the blue. So hyperblast is the yellow bit, and those follow the same fate as we see in birds. They'll end up undertaking gastrulation and neurulation in a similar way. But the outer trophoblast is the interesting bit. Components of some extra embryonic mesoderm and the hyperblast and the trophoblast are all going to go and form a placenta, and that placenta is all about making a blood interface between the baby and the mother, so that the waste can be exchanged, respiration can be occurring and exchanged, and also that nutrition can be passed. So we don't need an allantois, a chorion and a yolk sac per se, but we do retain the amnion, which is visible there around the baby as one of the extraembryonic membranes, and the others all combine to generate the umbilical cord and the placenta. So that is the story of vertebrate development.